Okay. Hello, everyone. My name is Lian, and I'm a master's student at University of Windsor. And today I'm going to share my research work about the cell type prediction using single cell RNA sequencing, and the techniques is the graph neural networks. So, as the name suggests, the single cell RNA sequencing allows the transcriptome analysis at the single cell level. And that being said, the cells will be isolated at the beginning of the workflow using the techniques like the enzymatic digestion or the mechanical dissociation. So the 10x genomic workflow, as the picture shows, is a typical method to process a large number of the cells and provide the valuable insight into the heterogeneity of the complex biological samples. So converting the gene expression data to a count matrix is a fundamental step in the analysis of the single cell RNA sequencing, as the count matrix captures the expression levels of the gene, the level of the gene in the sample, and enable a wide range of the downstream analysis. But the question is it possible to further convert count matrix into a graph? The answer is yes, because the graph is a data structure concept that used to represent the relationships between the objects. And in our case, converting the single cell RNA sequence data, the single cell RNA sequencing count data into the graph involves using the cells as the node and their relationships as the edges, which can be defined based on the various criteria such as the gene expression similarity or the co-occurrence. And as we've seen, the graphs encode rich relational informations. But the traditional machine learning techniques often just struggle to efficiently capture the intricate patterns present within these structures. And here's how the graph neural network step in and offering a revolutionary paradigm shift in how we can analyze and interpret the graph data. And unlike the traditional neural networks that operate on only the grid-like data such as the images or the sequences, the GNN directly operate on the graph structure data, allowing them to capture the dependencies and the interactions between the entities. The picture below shows the three main tasks where the GNN are mostly commonly used. So and to apply the GNN on a graph, Heterophily and Heterophily are the two most important properties need to be considered. And these two properties are opposite, and they are used to describe whether two linked nodes share the similar properties. For example, in the homophily, people who have similar interests are willing to group in the same are willing to form a group in the social networks, and we can see it in the graph here. So and on the so the nodes of the same colors, they are clustering together. But on the contrary, in the heterophily, the connections between the nodes are mixed with the colors. So recently, most studies have put forward ideas to make GNN better at understanding the heterophily. However, there's uncertainty about whether this suggested method actually work well with the real-world dataset especially in the single-cell RNA sequencing domain. And the current data set in single-cell RNA sequencing cannot be directly used for the GN training, as they are designed for the clustering and other downstream analysis. And it is not clear that how to feed the single-cell RNA sequence data into GN models. Therefore, our goal is to propose a pipeline to construct graphs using the scRNA sequence data and integrate graphs into GN models, and finally predict the cell types. So here's the data set we used in our study, which is from the Gene Expression Omnibus repository. And the samples of the data are shown on the right side. So this data set consists of 4 human pancreatic islets sample and 2 mouse pancreatic islet sample. So they all have the 14 types of the cells. So in the table, the column represent the genes and the row represent the cells, and the value for each cell represent the UMI filtered counts per cell detected in the raw data. And this is entire pipeline we proposed containing 3 main steps: pre-processing, graph construction, and model training. So in the pre-processing step, we perform quality control to remove the high expression level of the mitochondrial genes, because they are often considered low quality or the stressed cells. And also we remove the outliers by observing the large numbers of the detect genes, as well as the high total counts in both scatter and violin the scatter and the violin plots. So, and we apply the normalization to remove the technical variation, and at the same time to keep the true biological variation. So the method we use for normalization is total count scaling, which means we normalize each cell by the total number of total counts over all the genes, and so each cell will have the same total counts after normalization. So in the feature selection, the highly variable genes are those that display the substantial variance in the expression level across the samples. By identifying and prioritizing these genes, we can focus on the most informative features for distinguish between the groups or the classes in the analysis. So the graphs are carried out by measuring the similarity between nodes using UMAP. To construct graphs with various homophily ratio, we change the combination of the distance matrix, for example, the cosine and the Euclidean, and also the number of the neighbors. So we plot the graphs with 5 levels of the homophily ratio, and we find that with the increase in the homophily, the clustering boundary of the cell group becomes more clear. And after the graph is built, we extract the address from the adjacency matrix to form an edge list, and we use the edge list to fed in the model training. Okay, in the model training step, the input data format is called Planetoid, which is the graph-based semi-supervised learning framework, and which has been widely used in GAN. It contains 8 files, and they can be categorized as the graph structure, the node features, labels, and the training testing split. So as to the result, we conduct experiment on 7 models, including the standard GNN, such as the GCN, graph search, GAT, and mixed hop, and 2 extra baseline models who are addressing the heterophily, the H2 GCN and GBK GNN. So in the table, you can clearly see that the GBK GNN and H2 GCN. Okay. So the result of the GBK GN and H2 GCN outperformed than other GN models, including the MLP, especially when the H ratio is pretty low, like in the 0 to 0.2, and which means the heterophily is pretty high. Okay, in the summary, this study discovered that while the conventional GNN models such as the GCN or graph search, GAT, and mixed hop typically presume the homophily in the biological networks, but this assumption doesn't always hold up. So the GN models such as the H2 GCN and the GBK GNN has been specifically developed to acknowledge that the heterophily can be prevented in the specific single-cell RNA sequence data set. Okay, for the future works, we plan to tune the hyperparameters in the baseline models and maybe design a new GN models that are dedicated for the single-cell RNA sequencing based on the current baselines, and also to see if we can predict the connections between the cells, which is the edge prediction. Okay, and so I would like to thank you to the co-authors for their valuable contribution to this research, and as well as the sponsors. Thank you.
Questions? So I had a quick question.
Sure.
The results slide, what were those numbers?
Oh, they are the accuracy, actually.
Against a ground truth of cell type annotations? What's being evaluated?
So it's just the accuracy, like the true labels divided by the the true prediction divided by all the.
So the true labels here are of the cell types of cells.
Cell types, yes, cell types.
Okay,